Hi, I'm Somesh Mohapatra, a PhD candidate in the lab of Professor Rafael Gómez-Bombarelli in the Department of Material Science and Engineering at Massachusetts Institute of Technology. Today, I'm going to talk about my work on vaccine design using machine learning of human Degrons. This work was done in collaboration with a lot of folks. I have been the lead computational author for this work, but a whole lot of the work has been done with the help of experimentalists, being led by Nicholas L. Truex, who is also the co-first author on this paper. In terms of the labs, we were fortunate enough and grateful to collaborate with Professor Darrell Irvine, Professor Bradley L. Pentelute, and being advised by Rafael Gómez-Bombarelli at MIT. For this specific talk, I'd be going through the background and challenges, essentially introducing the problem and why it is so challenging to develop vaccine peptides for cancer in general. Then our approach as to what we hypothesized and took in terms of the computational efforts and the experimental efforts to address this problem. And finally, going to talk about experimental validation of how we validated the model and a quick summary of the entire work. Starting with background and challenges, the what and the why. At first, vaccine peptides or peptide vaccines follow the approach of cancer immunotherapy to target cancers. The therapeutic approach starts with vaccine target and characterization, then following with antigen selection and production, vaccine administration, and then waiting to see immune activation, that the body's immune system takes over and then kills the cancer cells in the human body. At this point, I'll walk you through all of these processes, starting with vaccine target characterization through immune activation as a part of this background. So at first, either the DNA, RNA, or the protein is taken and sequenced. Essentially, a part of the tumor is acquired, which is essentially called as biosample acquisition, usually by biopsy, and characterized by sequencing. What you see here is essentially a sequence or a list of sequences of amino acids that were found on the proteome in the tumor cells. And once you see this, then moving on to the second step, the mutations in bold and red are identified by comparative sequencing. So essentially, a part of the tumor genome or the proteome is taken and that is sequenced and seen that, okay, so this is what the sequencing looks like, or this is what the sequences look like. And then the same thing is done with things in the human genome. And when they're compared together, it is found out that, okay, there are things which are unnatural or not present in the other cells in the body. So these mutations might be the cause of cancer. And once these specific things have been found, what is done is the vaccine peptides are designed essentially starting with 25 MERs for immune memory responses. The 25 MER peptides are essentially required for these peptide vaccines. The idea is to generate some CD4 plus T cell responses. But then after further degradation, which is essentially we would want smaller epitopes as well, which are the desired epitopes or 9 MERs for cytotoxic responses in CD8 T cells as well, after intracellular processing and presentation of these vaccine peptides. In general, however, it is not known as to what these small 9 MERs are and how big these 25 MERs would be. In terms of a quick understanding of what the terminology is and what these sites are known, this is how a typical peptide vaccine looks like. At the center is this epitope, which is essentially going to incite an immune response in the CD8 T cells, and flanking that particular epitope on both sides are the N-term amino acids and the C-term amino acids. Now, it is not known that how many flanking amino acids should be added. Thus, the 25 MER peptides are generated by predicting this 8 MER or 9 MER epitope, but then adding random amounts of flanking amino acids onto each side, usually up to 25 amino acids in length. Even though the total longer peptides are difficult to synthesize and dissolve, they're still used. However, most of them do not usually lead to a therapeutic response. Now that the vaccine has been like developed and administered, we move to the space of characterizing, like whether there was an immune activation or not. However, it is really hard to go and like track all the human subjects that like, or the clinical subjects that were administered the vaccines in real time, and a lot of the variations that we would really like to have in developing the optimal vaccine is not really possible in clinical studies. So we take a step back and try to understand or approach vaccine design by studying the stability of the human proteome in general. So here, what I'm showing are like figures from two distinct works done in the labs of Professor Stephen Elledge, where they started with an oligonucleotide synthesis, performed PCR amplification, and then developed an oligonucleotide cloning into the lentiviral GPS vector. And when they were putting this into the particular assay, they sorted the cells, which showed that these particular proteins were essentially degraded by the cellular mechanism, which is essentially an ubiquitin and proteasomal degradation mechanism, or not. So what this essentially shows us that across the entire human proteome, there are proteins or peptides which degrade more versus which degrade less. And why this is important right now is the challenge that we see in the vaccines. The hypothesis that we are starting here is that if a particular degron or the motif at the end of this epitope on either the N term or the C term is not present, then there is a minimal processing of this entire peptide vaccine. And that leads to like a lack of therapeutic response in terms of the entire vaccine administration and workflow. Our hypothesis and the proposed solution is that we want to ensure that this particular degron sequence or a degron motif is present either at the N term and or the C term, or even both if that's like possible and if that's feasible. And when that degron is present, we want to have efficient and optimal proteasomal processing of these intracellular antigens and harness the natural machinery that the cell has, essentially the ubiquitin proteasome system, using these human degrons to facilitate how antigens are processed and presented to the immune system in the body. Now, moving on to our approach, and what we try to do here is we try to learn as to how these peptides are essentially being presented to the cells and how presented to the immune cells in general. And that is by machine learning of human degrons. At first, we start with two distinct datasets from the works of Steve Elledge, which I essentially shared earlier, with modifications at the C term. If you note, let's say sequence 2 and 3 noted here, the entire sequence is similar except the last two amino acids, where it is changed from KN to RS here. And even in this case, where it is for the N-term degrons on protein stability, if you notice, let's say these two sequences, again, 2 and 3, the entire sequence is same except the addition of this methionine group or like methionine amino acid at the earlier end. And by this, they essentially looked at global protein stability, understanding as to what is essential on getting these proteins being stable across the human proteome, and dividing it into like N bins, sometimes 4, depending on the FACS assay or the flow cytometry cell sorting assay, or 6 bins, depending on how the assay was conducted, to get like how stable they were. But at the same time, these are just probabilities of the protein being present in cells in any of the particular bins. So what we did is moved into trying to get a single number or a CDI, a C Degron index score, where we try to take all these probabilities, let's say peptide YY until NA is present in all of the bins with a high probability of 0.62 in bin 4, which, as the assay suggests, it's a very stable protein. And we try to look at that, essentially knowing that, okay, we try to have an exponentially weighted linear combination function that can provide a single value that indicates relative stability. So we got this value of CDI, which is like multiplying each of these bin values with an exponentially increasing coefficient and getting a singular score for all of it. Now, higher CDI is going to be a more stable protein candidate, and a lower CDI is going to give a less stable protein candidate. Now, with this data set where we had a list of sequences and a list of corresponding CDIs and NDI scores, we wanted to develop a machine learning model where we wanted to go for our work on sequence representation and learning from earlier. So this is something taken or like inspired by a paper that was recently published in Nature Chemistry, where we went on to represent the amino acid as a circular fingerprint and the sequence essentially as a sequence matrix with these fingerprint bit vectors arranged in the same order as they would be arranged on a peptide backbone. Using this representation, which is essentially very generalized and global for all sorts of sequences and can even take amino acids that are not present initially in the training data set, we try to use this representation to learn the CDI and the NDI. So this is the general model framework that we have, where the input is the fingerprint matrix, and we are trying to learn the CDI and the NDI. And dividing and splitting the entire data set as 60, 20, 20 as 60 as trained, 20 as valid, and 20 as test data, we got really good results for a particular model architecture, essentially a CNN model, where we had an RMSE of 12.57 on the absolute scale and an RMSE of 9.02 on the absolute scale for CDI and NDI respectively. Moreover, the RMSE values on the relative scale or the unitless RMSE values were around 0.3 and 0.4 on each of these cases, essentially ensuring that the machine learning models were able to predict CDI and NDI for the test set with reasonable accuracy. We also performed a whole set of benchmarks where we went for MLPs, logistic regression, linear regression, XGBoost-based models using this fingerprint matrix representation and one hot encoding representation, predicting the output with the CDI and the NDI. Now, before we move on to the experimental section, we faced a very unique challenge, and that challenge was, are CDI and NDI like related, or it does not even matter? When we plotted the predicted CDI and NDI for a set of 30,000 random sequences, we observed that the correlation between the Pearson's correlation between predicted CDI and predicted NDI was pretty high, with a Pearson's correlation value of 0.85. Thus, to ensure that we can modify like one particular variable at a time, we fixed the NDI and picked sequences across the entire spectrum of CDI for the experimental validation. Now, moving on to the second part about experimental validation, we wanted to tailor the antigen processing and presentation here. To do this, we used an anthrax delivery system that is very well characterized and used widely by the Pentelute lab folks, and Nick was leading the work on the experimental validation here. To keep things simple, essentially, we tried to take a set of about 7 distinct peptides that we wanted to try out. And in this entire set of sequences, we wanted to see that there's this particular N-terminus glycine, which would facilitate rapid degradation. And we wanted to see as to how the cytosolic peptide delivery would be affected by the C degrons, essentially with the epitope as synfical here and adding one amino acid at a time on the C terminus. We wanted to see as to how that is going to affect the immune response or even the protein stability at first. The first task was CD137 plus and CD69 plus. Both of these data refer to early activation markers on the T cells, but the earlier point is too soon for the T cell proliferation analysis. So after 72 hours, the T cell proliferation data shown here refers to the same T cells, but the T cell proliferation is complete. So this is essentially like the first data is to see that whether we are moving in the right direction or not. And this is the final data that you see here. In like across every single data point here, we see that the proliferation is perhaps greater than 75% or more or less greater than 75% across all of these data points. One thing that we noted here is that like the CDI becomes more relevant when we have a lesser concentration or a lesser dosage that is given into the cell. So you can note for the gray values here, the 0.1 micromolars, it is more relevant or like more distinct across different peptides, while for the reds, it is hardly mattering as to which peptide we are trying to analyze. To look like more closely into it, we plotted the Pearson's correlation coefficient. We plotted the percentage divided, essentially the T cell proliferation and the predicted CDI across the different concentrations or the dosages. And as we had seen in the bar plot earlier, here also we observe that the Pearson's correlation at a lower dosage is significant with a 0.81 PC, and it decreases as we increase the dosage. In summary, we started with vaccine administration and challenge. We proposed a hypothesis that harnessing the ubiquitin proteasome degradation machinery that is present in the cells, we can tailor antigen processing and presentation, thereby affecting immune response by that process. We used data sets on experimental global protein stability, and then used the approach of getting CDI and NDI scores, finally training an entire model framework on the sequence matrix representation, learning over CDI, NDI with reasonable accuracy, selecting sequences with a fixed NDI and across a whole lot of CDI, then finally seeing that our hypothesis is validated in vitro experiments. That is all from my side on vaccine design using machine learning of human Degrons. I'm happy to take any questions at this point. If you're watching this live, feel free to ask my questions once this conversation opens up, or leave your comments in the YouTube comment section. Thank you.